Hello, everybody. My name is Elsa, and I'm here to welcome you to this short series about citizen science and the Dive-In project. Today with me we have Leon from Slovenia, and we're going to talk about the DNA extraction. So thank you for being here.
Thank you for having me.
Thank you. And can you just tell us a bit more something about you, Leon?
Yeah, I can. Don't worry. My name is Leon. I come from Slovenia. I am a biologist by education, currently working as a professor of biology. So yeah, I have a lot of passion for biology. I come from a background where I've read a lot of biology and everything, and I really enjoy biology. That's why I said I wanted to be a teacher or a professor, so I can, put that biology knowledge forward to the people to make somebody as excited about biology as me. So yeah. That's me in a nutshell, but I'm also a little bit chaotic, so I apologize in advance.
No worries. We love your attitude. So, what is citizen science for you? As a biologist, as a professor, what does it mean, the topic of citizen science?
So, citizen science is a very fun concept for me, because I think that a lot of people have many times before shown that they don't really like science if they don't understand it, right? And that's usually the problem about big concepts that are sometimes or very, very sudden, and they flow into the general public, and people don't really know what to do with the words, don't know what to do with all of the data that just suddenly surrounded them. So, they start to lock themselves away from science. And I think citizen science in that concept where you can really put that science into more of a better perspective for people to understand it, or at least make a very good introduction to science for people. And I think that citizen science in that part can do a lot of good instead of bad. One good example was a lot of people, at least back at home in my country, had a lot of problem understanding the situation about quarantine and everything, and all the words about viruses and everything. So I think that if we would approach them a little bit more subtly, not like this big boom of scientific words, big words, nobody understands them, and they don't really know, so they think, oh, you must be lying. Because, it's very obvious that somebody's lying if they're just spitting words at you that you don't understand. Okay, so they must be lying. And it's not something that they do on purpose. It's just a normal mechanism. But if we can introduce some of the concepts a little bit better, then they'll say, okay, yeah, I know something about that. I might look more into it myself. And when they start looking into them by themselves, that's when they start to, get that general thinking about knowledge, positive thinking about knowledge, and positive thinking about science as well, because science does really help us with a lot of things in our everyday lives.
Of course, I totally agree. And sometimes we just forget how important to start from the small things, the very easy concept to go on. So we need to build from the very beginning good knowledge about science in an easy way, right?
Definitely, yeah.
Because science can be scary in a way for normal people, not for people maybe just like you.
I mean, it can be very scary for scientists as well.
So
it's this fun world of the more you know that you don't know that much.
Oh yes, it was just like that. It's true. Can I just ask you, maybe if you remember the moment or the people made you passionate about science?
I do remember. I remember a specific professor. So in Slovenia, the general schooling system goes through primary schools, which is 9 years, and then goes into the middle school, which is 4 years. And after that, you can, if you chose the professional route, you can go work, or if you chose this basic gymnasium route, you can go forward onto college. I remember my middle school professor. I was visiting gymnasium, and it wasn't something about her. It was just more of her approach towards teaching. She didn't teach like everybody else. When we had to come into the class, stand up, be quiet for the whole 45 minutes, and just be there, exist, and just listen to what she says. It was more of a dual communication. She was speaking to us, she was asking us questions, and the most important thing that I think I am doing quite well now as well, because I wanted to learn it that way, is how she used the concept that was pretty hard to understand and put it into, you know, normal words with a metaphor or something like that. You know, how a butterfly can fly, instead of saying, oh, you know, cytokinesis or mitosis or something like that, you know. And she made us understand things by comparing them to everyday life, which is also in essence citizen science, right? Yeah. So it was her approach that really got me into this. Okay, so science isn't that bad. It's really exciting because you can find out a lot of things. You can go and just put everything and make it yourself at home. It's not that big of a deal. You just need to have some proper pre-knowledge. And that pre-knowledge is what she gave us. And I'm, to this day, still thankful to her. And I still credit her for being the proper, you know, the proper idol for me to start studying biology, to really go into biology, because at that point I wasn't really sure what I was going to do. My mother always said, you're going to be a doctor. I said, I'm not going to be a doctor because I don't want to learn. But
you're learning even more.
Yeah, she really produced this new kind of thinking in me, saying, you know, you can do a lot of things that you want to do. You just have to be interested in them. And to this day, I still don't care about things that I'm not interested in. If I'm intrigued by them, I will listen to others. But the things that I'm interested in, I will dive deeply into it by myself, you know. First create opinion on your own, and then compare your opinions to others. Don't just stand behind your opinion, like compare it to others. You know, have a dialogue, have a talk, you know, and then your experiences, your knowledge will just grow naturally.
That's really inspiring. I really thanks her too, because I think that we should have more and more this kind of educators or professors just like that, with a lot of passion at the end that they want to share in a good way and an easy and dualistic way. So happy that you had that professor a lot. But let's talk about what do you do? Like what kind of project do you carry out to involve people in science?
So obviously, first of all, projects that I do like on a daily basis are projects with kids or students that I teach, that I am in contact with every day. And I just try to really get them to, you know, be excited about, not be excited about the subject, just be excited about researching something, knowing something new, be curious, you know, because getting curious is going to get you a lot further than just being good knowledge, you know, or intelligent. If you're intelligent and you're not curious, it's worse than not intelligent and curious. At least for me, in my concept.
Smart.
But apart from that, I do have some voluntary work that I do. I voluntarily help at a local youth center at where I live. We have a lot of general public that we can reach out to, and we can grab them together to come here and say, okay, so we have this concept that we would like to introduce to you. Do you want to help with it? And some of the projects that I mostly remember was where we were, me and another madam from a local botanical garden.
Okay.
Came in, you know, that botanical garden does a lot of different spices for food, salts, like really amazing assortment of every items, like really botanical, and they're selling it, and they're a pretty big thing. So, and on the other side, it was me, the student, and we were introducing to the population. We didn't really have like an age limit to from where to where you would need to be aged to come to that. It was like open for everybody. And I do remember that most of the public that came to that was mostly elderly people. But it's fun because in botany, or especially in the use of the plants that we can use every day, wild plants that we can use, you know, it's not something that is generally known, and it's not generally taught as well. Mainly for the reason because most of the data that we have today is just it's like mouth-to-mouth communication. It's not written. We don't have. Well, now we have a lot of books, but all of those books are based, especially on older people.
Yes.
That got their knowledge of that passed down to from their grandparents and their grandparents from there, and so on and so on and so on. So it's like this very elusive branch of biology where you don't really have that much scientific data, but you have a lot of cultural data. So it's like a really clash on, because doing science and everything usually means being very objective, while cultural data usually brings in subjectiveness. So it's like a really edgy branch. But we had this amazing time with all the people. They were asking questions. I had a PowerPoint about I think I had like 25 different plants on it. Different plants that grow outside of here, like very nearby, what you can use them from. And once you dive into that, you really know that some of the people know this, some of the people know that, but together in that room, everybody knew what I had, right? So it wasn't, for somebody, this plant was new, but for the other one, the plant wasn't new, but it was the other one that was new, and so on and so on.
Yeah, of course.
And then hearing back their experience, their there are different times where they made anything of their own or something like that. It came into it, and it came together like a very good project about plants and how we can use them. And, because some of the plants, we see them every day, we step over them, we pick them up, but we don't really know what exactly hides in that plant. And then some of the other projects are really good. Were like with when we had, if I directly translate it from Slovenian, creative schools, they're based on kids having a lot of time when they have holidays. So for the week during the holidays, we can give them different areas of expertise and how they can maybe do some projects, maybe do some workshops and everything like that. So we had a lot of that. And one time that is probably closest to my heart as well is when we were talking about cave ecosystems, or cave habitats to be correct.
Yeah.
And we went into that cave. That we have a smaller cave nearby, and it was like this really fun experience of kids really. They know Postojnska Jama, and I think a lot of the other who might be listening know where Postojnska Jama is, but Postojnska Jama might be a little bit too touristy at this point.
Okay.
But not for the wrong reasons. I'm just saying that you are probably unlikely to see a lot of the cave organisms that were living there, still living there, because they obviously need darkness, and if you want to go through the cave, you need light. So it doesn't really work that way. But when children saw, like the raw, natural touch, no human touch, nothing touristy, anything. We had to climb this forest to get to the cave and everything. It was like their happiness. Oh my God, look what I saw. I saw this. And then you have these cave spiders that are amazing. They have these little cocoons that are going down from the ceiling, and they were like, oh my God, this looks like a horror movie. Oh my God. And yeah, just seeing how projects like this on these topics can really make people have fun, be more curious and everything. I think that with everything that we've done throughout the years, I think that not a single one of them went over anyone's head. It was really good. I think that these projects are made, and I still do hope that I will be able to make a lot of these projects in the future.
I'm really, really happy to hear that. Thank you. So, Leon, what is the future for citizen science for you, or with citizen science?
Me involved in citizen science, or citizen science in general?
Also, maybe with you involved. Also, only the topic of citizen science, the concept of citizen science, how will it grow or evolve?
I think it's a very good concept, and it has a lot of potential. It just needs to be introduced the right way.
Great.
It's a dangerous area, speaking to people who don't want to be spoken to about science. It's a very intriguing task, and also pretty difficult to execute. The only thing that I would say is a lot of different people can be reached by citizen science, but we shouldn't and we must not believe that we will reach everyone. That is, I think, the most important thing. But otherwise, it's a very good concept. I really like it. I really enjoy working with it because I like introducing people, not just to science, but in general, I like teaching somebody something new. You know, it's like a good day goes by for me if I taught someone something new in that day, right? It can be a small thing. I can, why is, I'm going to have the most amazing, example. Why are the cigarette boxes in Slovenia that color? Because scientists discover that color is the most or the ugliest color in the world. The color doesn't even have a name yet. Not just in Slovenia, I know the color of every cigarette box that was recolored into one singular color.
Okay.
But it's the fact that they went into research to find which color is the ugliest so people would stop smoking. And they found that it was this color. It's like this poopy gray, it just, but yeah, the reason behind it is because the scientists went through and said, oh, this is the ugliest color.
Let's do that.
Yeah. And now you think about it and you say, okay, so if I don't understand the whole concept, because I don't either understand how were they comparing colors, maybe by use or something. But if you go through it and see, if you go through the whole article, I'm not going to explain because I don't really understand it yet that much. But if you go through the whole article, you see that it's a lot of different big words that you don't understand. And at the point, okay, so just they made this color because it's ugly. Okay, cool. But if I go deeper in and I go, okay, so what does this mean? This mean? I can see, okay, so now it does have a reason why it's called the ugliest color. And I'm not going to tell it. You have to research it on your own. Okay. Because you're going to participate in research now.
Homework. Okay.
Yeah, but no, it's, examples like this make it fun. Examples like studying for I don't know, for being asked in school or whatever different reason you have to study it for, it's not fun, right? It only begins to be fun when you study it for yourself, not for somebody else. And citizen science, I think, gives us just that, and it should do well. It should do well. I hope it will do well. I am really excited for it to do well, and I really hope that I myself will also be able to participate in it more.
I wish you good luck for that, but I'm sure that it will be. And I'm going to go with the last question, but I know that you also had something for us later to show us something. Mm-hmm. But how would you explain how can be useful or important for the people to get involved in citizen science?
Just like I said it in the beginning, I'm going to use the same example. Because it's like a very nationwide and very well-known example of the disease that came upon us in the year 2020. And I think that in these cases, we can really see how helpful it can be to have just the basic general knowledge, not like very specific. You don't have to understand what mRNA is or whatever other, or proteins, or what the proteins do and how our virus is made. You just have to understand the basic concept of how they're transmitted, or why do we wash hands? Why would we need to wear a mask? And et cetera, et cetera. I'm the one to go into any specifics or any, gray areas. But I think that if people refuse to do that, right, they should refuse to do that because of how they think about the science behind it, not because somebody else told them to refuse it. That's the thing I hate the most with every single concept that out there that it is. People who are hating upon it are usually the people who don't really have an idea what it does. And when you go Facebook comments, right, it's the best thing. When you go and you dive into those Facebook comments and you see that people, to me, at the beginning, it was funny, but now it's starting to get sad. People thinking about different things and just getting it completely wrong. It's not just messing with their understanding of that. It shows you that they might not have the great teacher in life that would show them the open-mindedness. And I think citizen science could help achieve that, or just overturn those, at least who want to be overturned, to really dive more into it, dive in. and just, enjoy life a bit more, because you don't have to be loud about every subject, but you also shouldn't be quiet about every subject. So yeah.
Leon, can you show us some experiments, maybe the DNA extraction? You bring something with you, right?
Yes, I brought a few things with me, and what we're going to be doing today is we're going to be extracting the DNA from strawberries. Why from strawberries? Because DNA, every single organism has a different number of DNA strands or DNA chains inside molecules inside of their cells. Strawberries happen to have a lot of those, so it's very easy to conduct this experiment that, by the way, you can all do at home. It is a very simple experiment. It only has a few steps. And you can see the DNA afterwards. That's why we have the wooden stick. So the wooden stick is also the first equipment that I bring with myself. That's, for example, I had to eat with forks in a Chinese restaurant just so I can bring this here.
So thank you for doing that.
Yeah, a sacrifice never to be returned. Next one is the strawberries. So I have some pressed strawberries here. Dish soap. Why dish soap? I'm going to tell you more about it later. Salt, some water that I have already mixed dish soap into, ethanol, 96% ethanol, and some Ziploc bags. Now, usually we would also need a coffee filter or a filter of any kind to filter out debris, but it can still work okay without the coffee filter and everything. The only thing that we're gonna also use it for is the cup. Okay, so first of all, what we're gonna do is we're gonna grab some strawberries in the Ziploc bag, and we are going to mechanically. Why I say mechanically, it's a very long-lasting joke of me saying mechanically, because it's mechanical force, I call it, if you smash by hands. So I would invite you to do that for me. Use the 3 strawberries, get the green part off so we don't get too much of it inside of the bag, and then put the strawberry inside the bag. Once you put those 3 strawberries in, you just smush them up. So what you need to make sure while doing this is that you do get some liquid out of the strawberries, and as well as not to have too big of a piece of debris inside or parts of the strawberry. We like those in yogurt, but we don't like them here in this experiment. After that, while you're still doing that, we're going to add a mixture called the extraction mixture or the extraction liquid. We're going to add it to the Ziploc bag. Now, what the extraction liquid does, before we say we talk about that, we need to dive a little bit into the normal or the general knowledge of a cell. Now, inside of a cell, DNA is located in the nucleus. In the nucleus of the cell, which is the main organelle of the cell. And nucleus and cells both have membranes around them. If we want to extract the DNA out of the membranes, what we need to do is we need to dissolve them or make them go away. Now, one part already does that is us pressing on top of these strawberries, because we already hurt some of the cells and the liquid comes out, etc. So we should already have some DNA free flowing inside of our Ziploc bag. Now, dish soap in combination with salt, what it's going to help us do is it's going to help us dissolve the membranes, because the membranes, as we all know, are made from fats. And fats are easily dissolved in dish soap, while they're not easily dissolved in water. Now, water is added only to make it a buffer. So I'm doing this by hand. I don't have an exact number of how much you should add, how much you shouldn't add. I would suggest you try it at home, and you'll see how much you need and how far you can go. Also, usually what you would do, you would mix the salt with the water because it makes it easier. Now, because I have it in such a strange package and I had to drive it all the way here, I'm gonna add it afterwards, so a little bit of salt. And now what we do is we mix up the bag so we properly get into it. We mush it a little bit more. Okay, so I'm gonna just mix it up a little bit. We'll see. Not my problem if it's. We're gonna mix it up a little bit so the salt gets dissolved in the water. Now I use the salt that is finely grained for this. You can use hard, or you can use not fine grained or just normal grained salt for it, but you're gonna need a little bit more time to dissolve it in the water.
Okay.
Because we do want it to be dissolved in the water. We don't want it to just be like an addition, but we do actually want for it to dissolve in the water, so we get the salty water, which then will take the effect, not the salt itself. Because the hard parts of the molecules are not going to be very effective if they're not liquid.
Yeah.
Okay, so I think this should be enough.
Okay.
I think we'll see. Now, also, this process is somewhat shortened for the podcast as well. But just so we know, usually what you would do is you would add a coffee filter so you can filter out the debris. Now, why filtering out the debris is better? Because you separate the DNA, which is located now in this liquid. This liquid already helped us extract the DNA. It helps us remove all the debris that is not necessary to have there. It just gets dirtier and dirtier.
Okay.
But in this case, I can also show it without a filter.
It can work.
It can work, yeah. Okay, I think this should be enough. I'm not going to use everything. I'm going to use maybe half of it so we don't overfill the glass. And then you just pour it inside of a glass. Now I'm also trying to be... Oh,
nice smoothie.
it's a smoothie. It's a soapy smoothie.
If you like, though.
Yeah, I wouldn't try it if I were you. Okay, so now we have this inside of here. The last thing we need to add is ethanol. Now what ethanol does, DNA we know is a molecule, and every molecule has its own properties. I'm not going to go too much into detail because it's not really that necessary. But all we need to know is that DNA just helps us separate the DNA from the water into the ethanol.
Okay.
So we slowly pour the ethanol into the glass.
Okay.
And what we should be able to see on top of it, or on top of the whole glass, where the ethanol is now separated from the water, we should see this white strands starting to pile up.
I see.
Now, all of these white strands are DNA. So what you see here is DNA, and now you can use your glorified
I'm ready with my stick.
and you can try and pull some of it out.
Whoa.
And you can see that the structure is strandy, so it is.
It is.
Yeah, and it's also in the white color. Now, if you were to try this, I wouldn't recommend it now because it's salty and a lot of dish soap inside of it, and also pure ethanol. But if you were to try it, it wouldn't have any taste. It is tasteless in nature. It doesn't have any properties of a taste. But if I stir it up a little bit and maybe pile some on top of it, now you can see why the debris is unnecessary to have and why it's good to filter it out. But you can see that it is possible to see the
Look how
extraordinary
much
DNA.
I have.
Exactly. And now you can see that this DNA is actually it's not invisible to our eyes, even though the molecule is just only a few atoms big.
It's true.
But that's because it's so long. Now, fun fact, if you were to stretch out your whole DNA,
Yeah?
or from a normal human being, you would be able to wrap it around the Earth to the moon and back 6 times.
Lovely.
So that's how much
Wow.
DNA approximately a human person has of this glorified double-stranded.
That'll explain what's the DNA.
Yeah.
Wow. That's really interesting.
The basis of all
I
life.
could eat it, right? We can also bake it in a way.
Yeah, so what I've done in the past in some other workshops is I've added a little bit less ethanol, a little bit less of dish soap. You don't get the result as like very intriguing and very visual like this, but you are able to eat it, and you can put it on a baking paper in the oven. Add some sugar. Now, if you really want to go all into it, you can bake the cookies. And once you bake the cookies, you can add a little bit of DNA on the top and then bake it all together, and you can have DNA cookies. Now, DNA doesn't have, as I said before, any taste at all. It's not going to ruin everything, now obviously, but don't add too much of it, not because of the DNA itself, but because of the other things that are mixed together.
Okay, of course.
That helped us extract the DNA. So yeah, this would be the end of our experiment. Now, this is all safely to be rid of in the toilet. Nothing in there is how can I say,
bad for the environment.
Yeah, bad for the environment, or
pollution.
not bad for environment, but something that we don't already put into our
Okay.
toilets or
Great.
sinks.
Thank you so much for showing us these DNA extractions, and let's hope that we will able to have some other great experiments through the toolkits that we are elaborating. So thank you again for showing us this.
Yeah, well, thank you for letting me dirty your lab here. But yeah, I'm really happy that you invited me today, and I hope we'll see each other soon.
Thank you so much again, Leon. Bye.
Bye-bye.